Welcome to the lecture presentation for Unit 3, Part 1 for SC090, Foundations of Science for Herzing University. As we do each week, we start with a unit outcome slide. Upon completing Unit 3, students will be able to identify the fundamental components of the cell, describe the function of the organelles, integrate nomenclature or the naming conventions of cellular biology, and introduce processes of cell division. We move on to a kind of a generalized introduction and overview. This week we'll take the next step up the organizational chain and begin our examination of the functional unit of life, the cell. If you go back, the atom is kind of the functional unit of chemistry. Now we're taking a step up. We're assembling chemicals into the basic unit of life, which is the cell. We'll explore the key components of the cell and the roles they play within the cell, and begin to kind of tease what they do within a larger organism. We'll also look at some of the specialized components of the cell and how they impact the cell's activities. Before we get too far into this week's presentation, I'd like you to reserve some time this week for a very special external activity. Your classroom contains a link to a game called Cellcraft. Please invest some time in this activity. I really, truly love this game and wish I'd had something like it when I was learning cell biology. It takes a few liberties with some real scientific facts, but you can't deny its value in learning about the cell. Several of this week's exam questions will come directly from the experiences you'll have in Cellcraft. You need to play the game in order to get a really good score on your exam. I think they also help you learn some important lessons about the cell. Now let's talk about the cell itself. The cell is known as the basic unit of life. Anything less complicated or smaller can't be classified as life. It isn't alive. At that point, it's just chemicals. There are a couple of notable examples, specifically viruses and prions, but they get included as life because they have a defined life cycle and hijack the cell to accomplish vital components of their necessary activities. So they're in that transitional space. Cells are self-regulating, meaning they kind of have rules that they play by and try to stay within those rules. They're capable of reproducing themselves, because again, if it's alive and doesn't reproduce, that ends the chain. And cells also have defined activities, non-random normalized activities. We see specialization in different cells as part of their life cycle and regulation. But nearly all cells we'll study contain the so-called big three. And this is a holdover from chemistry. We like to group important things into, the bigs. Last week in chemistry, or last unit in chemistry, we had the so-called big four. This week, within cellular biology, we've got the big three. The cell membrane and the cytoplasm are non-negotiable. Everything that's classified as a cell is going to have a border and some stuff within it. We also see a nuclei, but there are some specialized cells, like red blood cells, which will eject their nuclei. But at some point in its life cycle, every cell is going to have some form of nucleus or control center. And again, red blood cells, as the specialized cell, will eject their nuclei, but they've got them when they're younger. The cell membrane serves as the cell's outer margin, fence, and primary defense system. You got to have a border to separate self from environment. The cytoplasm is the cell's body. It's all the inner stuff, functional hardware, and a lot of water that's within the cell. The nucleus is the control center of the cell, holding the genetic material and supervising cellular activity. So let's jump over to our whiteboard and just do a fundamental drawing. So out here we have, let's click, okay. Out here is the world, or the environment. So this is the cell's world as a whole. You've got to have a membrane to separate you from the world. That separates self from world. Then you've got some stuff inside the cell. And I'm using blue for those of you who aren't colorblind. My apologies to any who are. That's the cytoplasm. And cyto as a prefix means cell. And plasma means gel or goo. Then you've got the nucleus. And this is where the genetic material is stored. This is where the instructions are established. This is what controls the cell's interaction with the world. It sets the tone. It's the boss. And we'll get into some further examples. But if you want, pause the presentation here and take a look. The three key components: cell membrane, cytoplasm or cell plasm, and then the cellular nucleus. So let's run you through just a quick primer on the membrane, because really this is the transition from world to self. You know, you have to separate the self from the world, the cell from the world. And let's start with the first thing we'll encounter. The membrane is actually a double-walled layer of special molecules. And again, we're going to carry over some terminology from chemistry here. This special arrangement is called a phospholipid bilayer, and we'll draw some pictures about that. The phospholipid bilayer provides a watertight seal for cells while preserving a way for chemical agents to enter and exit the cell. The specialized outer wall can examine outside agents and determine if they come in. This examination can take into account size, shape, electrical charge, polarity, that balance we talked about, before letting them into cells. And it is selective. It isn't random. The membrane has specialized ports and openings to allow passage of approved or desirable items into and out of the cell. The cell membrane is both tough and flexible and can even provide for a cell's movement. You want to pay particular attention to this in cellcraft. So let's jump back over and draw a membrane. So what we're going to do is we're going to take a section of the membrane and blow it up. So let's give ourselves a clear board. And again, we're over here. This is the edge of a membrane, and we're going to blow it up. So what you have, this is a phospholipid. So this is our friend phosphorus from the periodic table. And then these little tails are lipids, little fatty tails. And what we see is they line up like this. And I'll explain the reasoning for that. But it's a phospholipid bilayer, so it means there's two layers. Now imagine this all the way around. All the way around this cell, you know, you've got these tails both inside and outside. And I'm trying to do a fairly representative job here. What you've got is, and I want to introduce some terminology here, you've got hydrophobic and hydrophilic. And we're going to split these guys. What does hydro mean? That's the easy one. Hydro means water. Phobic means fear, right? If you have a phobia, you have a fear. Now, philic, here's a trick to remember this one. Philadelphia, big city on the East Coast. It's known as the city of brotherly love. Philly is kind of like feelings, so love. So hydrophobic is water fearing. Hydrophilic is water loving. So we've got our phosphorus, we've got our lipids. What do we know about fats? Because lipids are fats, right? Oil and water don't mix. So the inner part is hydrophobic. The inner part is hydrophilic. So you've got this transitional layer of likes water, hates water, likes water. You've got the outside of the cell. You've got the inside of the cell. So the world outside is usually not too dissimilar from the world inside. We like to be close in terms of our environment, but this wall gauges what comes in and what goes out. We mentioned how the wall can be selective. So the cell membrane isn't just this phospholipid bilayer, but there can be some ports in there or some tunnels. Sometimes there's proteins in the wall, and they have chemical sensors, we'll say, which if something nice comes along, it can grab onto it and move it into the cell. Likewise, if there is a chemical product that the cell needs to get rid of, it can grab onto it and eject it from the cell. Let's go back and talk a little bit more about the cell. We'll move on to the cytoplasm. The cytoplasm, or cell's body, is mostly water, but does so much more. And remember, cytoplasm kind of means cell goo or cell gel. It's the site of most cellular activity and typically forms most of the cell's bulk. It's a warehouse for hundreds of chemical agents, chemical ions, those positive and negative guys we talked about in the previous unit, enzymes, which are folded up proteins that help chemical actions run better, and different cellular products. The cytoplasm is a constant dance of cellular products, resources, water, waste products, invaders, and defense agents. There's a lot going on. It's a real busy compound. It looks like clear goo, but there's so much more going on. And we'll start to talk about what's going on in the subsequent slides. First up, inside the cell, we have the boss. The nucleus can be considered the main office or the control room. It's the hub, it's the boss of the cell. The genetic material or the instructions, the blueprints of the cell, are held in safekeeping here. In humans, this specialized blueprint is called DNA, deoxyribonucleic acid. DNA has different sections and zones which provide instructions for proteins and cellular components. A cell becomes who he is or who she is by following these specialized instructions. Different cells have the hardware to read different sections of the instructions, different part of the manual, the DNA, and make different products or react in different ways. The nucleus also holds a specialized inner office called the nucleolus. So this is like the office within the office, okay? The nucleolus is a dark spot composed of nucleic acids and proteins. It helps make little balls of RNA, ribonucleic acid, called ribosomes. We'll talk more about those guys in a little bit. Before we move on again, take a little look at the picture here. The purple, again, apologies to anyone who happens to be colorblind and struggles with the colors. This ball itself is the nucleus. Inside the nucleus, we've got all the genetic material for the cell. It's all the blueprints, kind of like they're locked away in a safe. The concentrated mass towards the center is the nucleolus. So that's like the boss's office within the business office within the factory. And that's where, the really important stuff is kept. That's where, the originals are kept, not the copies, the originals. And we'll talk more about ribosomes as we move forward in some of the other organelles. So now we've covered the big three. The cell membrane, the cytoplasm, and the nucleus. Now let's get to some of the others. These are the guys that lead to specialization. These are what help differentiate this one cell from another. They may not be present in all cells, but they play a vital role in the cells which house them. And don't forget, we can frequently tell what a cell is by examining the organelles which are present or are not present, if they're absent, and the arrangement of the organelles. Remember, organelle quite literally means little organ. You see it right there. Organ, first part, So it's the structure, and then L. We've seen that in other places, right? So L means little. So organelles are quite literally the tiny organs of the cell. Some students find it helpful to relate the organelles to the body organ counterparts. Like organs, organelles are specialized machinery of a cell, which with clearly defined roles and processes, got clear specializations and products. Personally, the example I like to use, I like to look at organelles as they relate to a factory model, and I've touched on it a little bit already. See the slide for my layout of the organelles. This slide is also a reminder of the value of Cellcraft in this week's studies. So again, if we look at the factory roles, the membrane is the outer fence, the security. It controls what comes in and out. The cytoplasm is the factory floor. That's where the machines are. That's where the manufacturing gets done. The nucleus, kind of like the foreman or kind of like the boss's office. There's a structure we'll talk about called the endoplasmic reticulum. That's the manufacturing line. That's a very specific machine for making stuff. The Golgi apparatus, that's the packaging department. That's where everything gets boxed up for export. You've got a structure called the mitochondria, which is the power station. Factory draws a lot of power, so you need to have a room or an area that manages the power coming in and how it gets used. And of course, like the bottom of the slide says, Cellcraft, 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 Cellcraft. Did I mention Cellcraft? You'll encounter all of these guys if you play the game, and really you can learn quite a bit in 15 or 20 minutes. Part 1 wrap up, and we're going to try to keep this shorter. I know chemistry was really detailed, but this is where chemistry becomes life. Before we got to this level, we were just studying chemistry, physics, math, boring stuff, nothing that was alive. Beyond the cell, that becomes a study of chemistry, physics, math, and life. So the previous rules still apply, but now we're applying it to life, living, dynamic things. We're outside the beaker, and now we're in the lab now. This week's lesson features a lot of terminology. Be certain you understand each term and are capable of defining, explaining, and citing examples for each term. Know what is and is not included in each term or process. This is a skill which is vital not only to this class, but your future studies as well. If you can't define, explain, and cite, then you haven't really learned it. You need to go beyond just harvesting information. I want you to also get out of a habit that I see a lot of students, and I'm kind of going off the notes here. But in future classes, I see students hunt and peck for words in a book. They'll see a question on a test, and they'll hone in on the key word or process in that question. They go to their book, and they read one line. That's good for extracting snippets, but that's not synthesis. That's not building knowledge. What you need to do is read the paragraph before, the paragraph with the word, the paragraph after. You need to understand the whole concept. There's kind of an old rule that I was given when I was studying, and that's if you can't explain it to your mom or your grandma, you really haven't learned it. So unless you can sit there and go, the cell membrane is made of these little round balls that have squiggly fat tails. The round ball likes water. The squiggly fat tails don't like water. So they line up and create this double-layered wall around the cell. And that wall creates a barrier between the cell and the world and controls what comes in and out. It sounds easy. And it really can be, but you got to invest the time. It's a lot simpler than we like to make it out to be. But if you can't do that, you haven't learned the material. So this is the wrap on part 1. In part 2, we'll advance our study, explore some common abbreviations, dig into some of that Latin and Greek again, and fill in more of the blanks. Thank you, and we'll see you in part 2.